Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International GCSE and 9-1 Biology, paper 2B for January 2022. This is the part 1. I will do this paper in 2 parts. So the first part will have question 1 to question 3, and the next part will have question 4 to the end. I will put the link to part 2 in the description box. So let's begin. So they say read the passage below, use the information in the passage and your own knowledge to answer the questions that follow. So here they say making enough blood for the world. A blood transfusion is a transfer of blood from a donor into a patient. Blood transfusions are routine medical procedures that save the lives of millions of people every year. We can see a pack of blood to be transfused. So they say the first successful blood transfusions were performed in the early 20th century after scientists discovered that there are 4 main blood groups. There is A, there is B, there is AB, and O. The blood groups are due to the presence of proteins called antigens on the surface of the red blood cells. There are 2 main proteins or protein antigens called A and B. If a patient is given blood with antigens different to their own cells, their immune system will make antibodies against the antigen. The antigens present on the surface of red blood cells for each blood group are shown in the table. So we have the blood groups here and their corresponding antigens. Continuing, they say currently blood transfusions are carried out with blood that has been donated by healthy people. The World Health Organization calculates that there are 118.5 million blood donations collected globally every year. Of these donations, 40% are collected from a small group of high-income countries. This means that there is a shortage of blood in many countries, so the hunt is on to find an alternative. Scientists have found a way to make artificial red blood cells. They made spheres of cell membranes filled with hemoglobin. So these artificial cells are then suspended in sodium chloride solution. These artificial red blood cells have no proteins on their surfaces. Another way of making red blood cells is being developed in the UK. A research team has used stem cells to produce red blood cells with blood group O. The red blood cells produced are then suspended in sodium chloride solution. Both methods produce large quantities of safe red blood cells. There may be other advantages as well. Artificial blood would always have the same concentration of solids and will not clot when stored. Critics have pointed out that our artificial blood will only transport oxygen and the blood has many more functions. So the first question here says, name the type of cell that produces antibodies. Antibodies are produced by lymphocytes. They are part of the immune system. Let me try to make this a little bit smaller. So they say, human blood groups are controlled by 3 alleles. There is the A, the B, and the O. The A and B alleles are codominant, and the O allele is recessive. State what is meant by the term codominant. So codominant means when both alleles are present in the genotype, their traits are going to be expressed in the phenotype together. Both traits are going to be expressed in the phenotype. The next part says 2 parents have genotypes A and O, so there is AO as well as BO. Ignore that I am just saying IA, IO, and B, IV, IO. So it's the same thing as saying AO and BO. They say which of these are all the possible blood groups of their children. So if we have 2 parents, these are going to be the genotypes they have here, the B and O and A and O. So that and that, this gamete and that gamete are going to produce this specific phenotype, which is AB. Here we have, you could say AO, or say OA, OB, and then OO, which is O. So here we have AB blood group. We have A, we have B, and we have O. So the answer should be a D, corresponding to the right thing we have seen. So let me try to make it not so big. Continuing on, they say calculate the number of blood donations collected per year from high-income countries, line 14 to 15. Give your answer in standard form. If you see line 14 to 15, we were given this value here. And again, I want to take you back just a minute, 14 to 15, line 14 to 15. We have this value here and that value here. So we're going to use these 2 to calculate what they're asking us to find. So let's go back slowly. it's going to be 40 because it was 40%. So 40 over 100 times that gives us 47.4 times 10 power 6. In standard form, it should be 4.7 times 10 to power 7. So that is the answer. Continuing on, they say some scientists have suggested that spherical artificial red blood cells transport oxygen less efficiently than normal human red blood cells. Explain why the shape of the artificial red blood cells reduces the efficiency of oxygen transport compared to normal human red blood cells. So I said artificial red blood cells do not have a biconcave shape. Of course, it's going to be hard for the scientists to make this biconcave shape. And as you know, the biconcave shape is very important in increasing the surface area for oxygen absorption or for gaseous exchange, basically. So here I say, so they will have a lower surface area to volume ratio. So less oxygen will come in or will be able to leave out. So less oxygen will be absorbed into the lungs and less will be released into the cells. So a spherical shape may prevent them from passing through the capillaries. This is another thing as well, because the biconcave shape we saw has qualities of providing a larger surface area for the absorption of oxygen, as well as allowing its shape to pass through the blood capillaries. So if it's spherical, that shape might prevent it from passing through the blood capillaries. Let's continue to the next part. Suggest why artificial blood does not clot when stored. So here we have to see clotting is brought about by platelets. So if artificial blood does not cause clotting, it means it does not have the platelets that are required to cause the clotting process. So I say it does not have platelets. Okay, so let's continue. They say explain why the artificial red blood cells are suspended in sodium chloride solution instead of water. Of course, these are animal cells, and if they're suspended in water, water is going to come into the cells, causing them to burst. So water will come in by osmosis. So to prevent that, they suspend them into a salt solution that could have a similar concentration or solid concentration like blood. So I say to reduce uptake of water by osmosis, because if they take up a lot, they might burst. Remember, animal cells do not have a cell wall, they just have a cell membrane. And the cell membrane cannot be good at preventing excess water from coming in by osmosis. So let's continue to the next part. Explain why stem cells can be used to make large quantities of red blood cells. This is line 22 to 23. Using stem cells, remember stem cells can rapidly divide, or they can divide by mitosis, producing so many more copies of themselves. And these copies have the ability to differentiate and become any type of specialized cells. So it means they can differentiate and become red blood cells as well. So I say it because they can divide by mitosis to produce more copies. These copies are of themselves, so like more clones. And then the stem cells can differentiate into any type of specialized cell. In this case, they can form red blood cells as well, because stem cells are not yet specialized. They have the ability to become any type of body cell. In this case, I mean stem cells. Of course, for IGCSE level, when we talk about stem cells, we usually talk about those cells that can become anything. But when you study biology at higher levels, you see the different types of stem cells. So in this case, I'm talking about IGCSE level. Stem cells can be anything, can transform or differentiate into anything. Still, when you use them, they can also make red blood cells. Continuing down here, suggest why scientists made red blood cells with blood group O. Now, blood group O, remember, doesn't have antigens, so there will be no A antigens and there will be no B antigens. It means since presence of antigens can trigger the immune system, so if there is no antigens, it means the immune system cannot be triggered, and the same blood can be donated to anybody since there will be no immune reactions. So I say it because there are no antigens on blood group O, so antibodies will not be produced by the lymphocytes, so any person can be a recipient of the blood. So this is going to be kind of like a universal donor. Next part says, give two substances found in blood plasma that are not present in artificial blood. There is no urea, there is no digested food, there is no carbon dioxide, no hormones, no vitamins, no antibodies, no proteins, no minerals, and so many things, because remember they told us sodium chloride solution, so we'll assume there is no mineral like calcium. So this brings us to the end of question 1. Let's move on to question 2. Question 2. Food items can often be spoiled if saprotrophic microorganisms such as mold fungi grow on them. Describe how a saprotrophic fungus such as mold obtains its food. Saprotrophic microorganisms feed on substances by releasing enzymes onto usually dead substances. The enzymes carry out extracellular digestion, and then the products from the digestion are absorbed by the organism inside its body. So here I said fungus release enzymes on dead or decaying microorganisms, and then the digestion takes place extracellularly, and then the fungus absorbs the digested nutrients into its body. So they continue to say, a student uses this method to investigate ways of preventing peas from being spoiled. They place 3 peas in each of the 3 test tubes as shown in the diagram, and then they cover peas in test tube A with water at 37 degrees. They cover test tube B with water at 4 degrees, and they cover peas in test tube C with vinegar, which is a weak acid, but at 37 degrees, and then they leave the peas for 24 hours. So this is the way the setup was done. You can see here test tube A, B, and C. So from this observation, we can expect that water at 37 degrees, we're going to find the highest growth of fungus. And then that at 4 degrees, there will be little growth. And in vinegar, we expect no growth because it's acidic conditions, and these acidic conditions will alter the pH, preventing the enzymes from being activated or from working properly. So maybe they could be denatured. So let's see the questions. The students observe the level of cloudiness of the solution to determine how spoiled the peas have become. The level of cloudiness can be used as a measure of fungal growth. The table shows the students' results. So we have the results in the table here. And as you can see, A, there is very cloudiness due to more fungal growth at 37 degrees in water. In B, just 4 degrees, there is slight cloudiness because this temperature did not provide the right kinetic energy for the substrate and the enzyme to collide in order for successful collisions to occur. And then for C, we see there is vinegar at 37, so there is no cloudiness because there was no fungal growth. So they say suggest a problem with using the level of cloudiness of the solution to determine how spoiled the peas have become. Now, if you use level of cloudiness, it means it can be viewed differently by different people whose eyesight is different or who are in different conditions depending on how the light strikes the test tube. So I say judging the amount of cloudiness is subjective. This means it depends on the viewer. Whoever is viewing it, they could have different results. So someone else could think that it's more cloudy than it is actually. The next part says, explain the appearance of peas in water at 4 degrees. So at 4 degrees, we have the temperature is kind of low, so there is less fungal growth. The reason for that is the temperature is not optimum, or temperature is below optimum, so the enzymes are not as active due to less kinetic energy. This will lead to less collisions occurring, so less collisions between the substrate and the enzymes will occur. And another thing, there will be less respiration to provide the energy for the increase in number. So we want to explain the appearance of peas in vinegar at 37 degrees. Now you remember from the table at 37 degrees in vinegar, there was no cloudiness. So we expect that it's because there was no fungal growth. So the temperature is appropriate because it's 37. However, the vinegar provides an acidic pH, so the enzymes are denatured. And then the denatured enzymes can no longer bind with the substrate. In this case, it's because the active sites have changed. So the substrate can no longer fit in. Therefore, the fungal growth is going to be lower, or it's going to be 0 due to the pH that has been altered. So this brings us to the end of question 2. I will move on to question 3. Question 3, the diagram shows a cross-section through a leaf. So you can see the leaf here. There is the upper, we have the cuticle, the upper epidermis, then we have the palisade, we have the spongy, as well as the lower epidermis. So they say, which layer contains the palisade mesophyll cells? It is this layer here, layer B. These have a lot of chloroplasts, as you can see. So it's B. The next part says, which set of environmental conditions would produce the fastest rate of transpiration from this leaf? So fastest rate of transpiration is going to occur if the conditions are windy. That is good, actually. So humidity, we need low humidity, and the temperature has to be high. So high temperature allows the evaporation to occur, or the water to be converted into vapor, and then
the low humidity allows for the stream to continue, or for the gradient to be continued, in order for more water to get out of the leaf into the surrounding environment. So it's going to be low humidity and high temperature. Let's continue to the next part. This scientist investigated the effect of changing carbon dioxide concentration on the density of stomata on wheat plants. So you can see they're investigating something. So carbon dioxide concentration is what they're varying. So that is the independent variable, and the density of stomata is the result, so the dependent variable. So they grow wheat plants from seed in different concentrations of carbon dioxide, and then after 3 weeks, they take a leaf from each plant and calculate the density of stomata. State the independent variable in this investigation. It should be the concentration of carbon dioxide, because this is what you're going to vary. So they say give 2 abiotic variables the scientists could control. Abiotic means non-living. So since this is an experiment about changing carbon dioxide concentration and then observing how the plant is going to react. So the temperature has to be considered, light intensity, mineral ions, pH, water, as well as humidity. So, and many more you could find. The next part, they say to calculate the mean density of stomata, leaf sections are viewed with a microscope, and then the number of stomata within 6 circular areas of the leaf are counted. The results of one leaf are shown in the table. So these are the results in the table. We have the leaf area number, and then here we have per number of stomata. We can see the results here. I will continue to the next page. Here they say the radius of each circular area is that. So they've given you the formula for area, which is pi r squared, and they've given you the value for pi. They say calculate the mean density of stomata on the leaf surface. So when they say mean density, so it's going to be stomata per millimeter squared. The mean is going to be, I added up everything you saw in the table and divided by 6. So I got 70 stomata. That is the mean number of stomata. And then I found the area, which is pi r squared. I have the pi and they have the radius. So I found that. So the mean density is going to be the area divided by the area, which is basically mean area, which is this one here. So we're going to divide the stomata, numbers of stomata, by the area, and we'll get the mean density to be that. So because you remember we said mean density is going to be the number of stomata per millimeter squared. So it's going to be 70 stomata divided by the area you got. So you get 140 stomata per millimeter squared. And again, you had to refer to this part, which was given. The next part says the investigation shows that in increased carbon dioxide concentration, there is a low mean density of stomata. The scientists conclude that in hot, dry areas, which is with increased carbon dioxide concentration, it would be an advantage for wheat to have a lower mean density of stomata. Discuss the scientist's conclusion. So, in this conclusion, realize that, yeah, it's fact, carbon dioxide is required for photosynthesis, so this is a fact, you can agree with him, and a fewer stomata would reduce the carbon dioxide intake. That is correct. However, since they are high, again, remove those one high, because they are high concentrations of carbon dioxide, uptake of CO2 may not be affected. No matter whether we have fewer stomata, provided we have a higher concentration, the uptake of CO2 may not be affected. So fewer stomata would reduce water loss, which is beneficial through transpiration, leading to wilting. So this would lead to wilting if we have excessive water loss. However, less transpiration leads to less mineral uptake from the soil. This is connecting to that. I hope you can understand. So fewer stomata would reduce water loss through transpiration, and this, if it was excessive, it would lead to wilting because of loss of water. So however, less transpiration leads to less mineral uptake from the soil, and again, minerals are taken from the soil through the water in the xylem. So if there is less transpiration going on, there will be less mineral uptake, and that will lead to problems due to lack of minerals like magnesium, which is useful in making chlorophyll, minerals like nitrates, which are useful for making proteins. So if there is less magnesium taken up, there will be a reduction in chlorophyll, leading to less absorption of sunlight. Less nitrates will lead to reduced amino acids, and then less transpiration will also lead to less cooling, so the plant could be affected. So you support the scientist in one way, but in the other way, you can also see the disadvantages of the method they're trying to introduce. This brings us to the end of this question 3, as well as the end to part 1 of this of this paper. So please do not forget to subscribe to our channel. I'll see you in the next video. Bye-bye.